Okay, today we're going to do most of, probably not quite get done with gas exchange. Then on Friday, we're going to start on nutrition and digestion. Next week, we'll do immune system function, and hopefully have some time at the end to do some practice problem solving. Okay, I wanted to remind you about respiratory gases and how they get from air to cells. Some numbers that you should remember are on here. At sea level, well, anywhere, oxygen is 21% of the molecules in air. Most of the rest, 79%, is nitrogen, and you don't particularly need to remember that, and a tiny amount, 0.2 millimeters of mercury, about 0.02%, something like that, is carbon dioxide. Water, of course, holds much less. So if we were drawing a water surface and we had oxygen above it, 21%, which would be rounding about 160 millimeters of mercury is the partial pressure of O2. Okay, 21% of the total, 760. And we had water molecules in here. The problem for oxygen is that anywhere there's a water molecule, an oxygen molecule, water molecules are attracted to each other, and so they have a tendency to eject oxygen. Okay, simply because oxygen does not interact well, doesn't form hydrogen bonds with water. So the result is you can have 160 millimeters of mercury partial pressure, gas tensions will be equal, and we often refer to that as 160 millimeters partial pressure of oxygen in the water, but in the water, at 0 degrees centigrade, that's only 10 milliliters per liter. Okay, at about 35 degrees centigrade, or 37, around your body temperature, only 5 mils O2 per liter. And that's because oxygen just does not interact well with water. If you think about why does it hold less oxygen as the temperature goes up, well, think about what's happening at boiling. So what's happening at boiling? What's happening to water molecules at boiling? Are they sticking to other water molecules? At boiling, the problem is you have some water molecules are moving so fast that they're escaping all other water molecules. They're forming a gas. So at boiling, you're even driving water molecules out. Guess how much oxygen you'd have left? It's all gone. Okay, so the hotter it gets, the greater the tendency to drive oxygen out. And the less oxygen water will hold. If we compared that to carbon dioxide, and I don't remember the exact amount, but 0.1%, it's something like 0.2 millimeters of mercury is the partial pressure for carbon dioxide. Very low level, highly significant because it's greenhouse gas, so we care a lot about it in other contexts, but there's not actually that much carbon dioxide in air. Again, if we had water molecules here, the thing about carbon dioxide is that carbon dioxide will form hydrogen bonds with water molecules. And in fact, water will hold about 1.5 times, actually more, carbon dioxide than the air above. So there's a hypothetical situation in this diagram. And if we're at somewhere around 35 or 37 degrees centigrade. Actually, this is cold water. Sorry, let me back up. So we're at 100 millimeters mercury, so this is a little less oxygen than you'd find at sea level. And in cold water, you'd have about 4.5 mils of oxygen per liter. But compare that to carbon dioxide. If you artificially put way more carbon dioxide in the air than there is, and you had 100 millimeters of mercury partial pressure of carbon dioxide, you'd actually have more carbon dioxide in the water at equilibrium. Okay. This is why you can carbonate beverages. Nobody sells oxygenated beverages, at least not if they're sensible. Okay, because the oxygen just leaves as soon as you open it up. But carbonated beverages work just fine. And you can demonstrate for yourself the fact that if you add heat energy, it will drive off those gases. So what I want you to do is get a can of soda, bring it back to your room, explain these principles to your roommate, shake it vigorously, adding heat, which will get the carbon dioxide to start coming out of solution under pressure, and then pop the top. Okay? Explaining to your roommate what's going on, okay? Barely even got chuckles. It's a good thing I only tell these jokes for me. Okay, does that make sense? Any questions? So toward the end of class last time I asked if people still needed explanations. Does that make sense to you guys? Any questions? Okay, the key takeaway is that it's hard to get oxygen into water, but animals need oxygen. So I mentioned path lengths. In the video. And so imagine we're in lung. And here's the air and the tissue side. Now remember, membranes don't tolerate completely dry conditions. So if you want to keep a lung exchanging oxygen and carbon dioxide across it, you need to have a wet mucus layer. So that's going to add a bit to the path length. Okay, if you make that mucus thicker, it adds even more to the path length. The more mucus you put in there, the further the oxygen has to go. And then it has to get across the cells. This is lung epithelium. So it's got a basement membrane. And then it has to get into a capillary. Which also has a basement membrane. And it's got some interstitial fluid to go through in between. So the path length includes all the tissue it has to go through to get to the blood. Remember that we have bulk flow currents bringing air into a lung or across a gill, bringing water across a gill. And we have bulk flow in a capillary through the circulatory system. So bulk flow is pretty fast. That does not impose any more delay than the time it takes you to get blood from one location to another. Still matters. One of the problems large animals like us have is it takes you a minute or so to get blood from your lungs, where it picks up oxygen, out to the tissues, maybe even a bit more than that. I don't remember exactly for an animal our size. For an elephant, we're probably talking 5 minutes or more. For a mouse, we may be talking 5 seconds. So a mouse is capable of delivering oxygen to tissues a lot faster because it's smaller, doesn't have to send its blood as far through bulk flow. So if we looked at an earthworm, with no lung, or I could draw, here's a flatworm. That may be only a millimeter thick. Diffusion alone will work. Okay, that's why a flatworm is flat. It has no circulatory system. It has no way to move material around by bulk flow. It has no lung. It's got to be flat. If you're an earthworm with no lung, you're pumping blood through this ventral vessel. So blood is going out like so, and then into capillaries, and back to that dorsal vessel. That network of capillaries right underneath the integument is where oxygen is being picked up. There's no lung. It's just a big gas exchange surface and a relatively low metabolic rate for these animals. So oxygen is being picked up by diffusion. The path length is whatever thickness of the integument is, the outer mucus layer, and then through the extracellular matrix to get to the capillaries. And so then oxygen is being carried back by bulk flow and then spread out to the rest of the tissues, including, this is the lumen of the gut. In the middle here, and that little tangle, this little tangle here is for the excretory organ. Remember the metanephridium? Okay. So that's sending blood to the excretory organ in order to get waste molecules and stuff into the excretory organ. Okay. So no lung, no gills, just surface exchange. I added a diagram here showing, just to remind you about the closed circulatory system of a worm, an earthworm. So how can an animal increase its oxygen uptake? What are the ways an animal can increase its oxygen uptake or increase its rate of getting rid of carbon dioxide? It can increase the flow velocity of the internal or external medium. Okay, if it's a fish, it can send water faster over the gills. You can breathe faster. Okay, and that'll increase gas exchange. You can increase the surface area for diffusion. Okay, you can have simply a larger total surface area of your lung. The lung structure you have in a human, somebody calculated it out. For a typical sized human, the lung structure is about the size of a tennis court, the surface area. Okay, so the surface area of your lung, if we spread it out, would cover, I don't know, half of this room. Okay. So you can increase the total surface area. That requires a bigger gill or bigger lung, takes more energy, more resources to do it. You can decrease the path length. Okay, you can decrease the path length. So if you make the tissues thinner, and I'll show you an example. I'm going to come back to this, but so here's a scanning electron micrograph of a mammalian lung. All the little lumps you see all over the place. So all these little lumps are places where there's a capillary. So here's a capillary with some branches. And the little lumps are where there happens to be a red blood cell right at this moment. So all these little bumps are red blood cells. You can see the thing is practically, the tissue is practically covered, practically filled with red blood cells, with capillaries. So lots and lots of blood flow. This is the opening into an alveolus, those little grape-like things where oxygen is being exchanged. So these are all parts of alveoli, and there's one opening, here's another opening, here's another opening. Okay, it's very thin. Here it is when we cut it in cross-section. So here's one alveolus passageways, here's part of one alveolus. Here's a capillary, these little things are red blood cells. Remember, red blood cells have a shape that is sort of like a frisbee, flat and thickened around the edges, right? So here are red blood cells cut through, and this would be a capillary. This probably bends a little bit, so this is probably the same capillary, but it's been sliced off here. Okay, and the path length, you can see, is incredibly short in a lung. You can't really even see it, but there's an lung epithelial tissue here. This really thin piece here is lung epithelial tissue. The membranes and the basement membranes are almost right on top of each other. So here's the epithelial, the endothelium of the capillary, which is right next to the gill. This is about the thinnest cells you've got anywhere. These red blood cells are about 10 microns. So the lung epithelial tissue is, in most places, thinner than even a micron. The path length across here is only in the neighborhood of about a half a micron. It's a very short path length. It's one of the reasons you can't afford to send blood under high pressure to your lung. Okay, remember in the circulatory system, we have a high pressure systemic circuit from the left ventricle, but it has to be a low pressure circuit to the pulmonary, to the lungs, from the right ventricle. It has to be low pressure because this is so thin it easily bursts. Okay. I mentioned racehorses that actually do have damage. They've been under selection to get their blood very rapidly through the lungs, so they have high pressure pulmonary circuits, and these very thin capillaries burst easily under those pressures, which is why after a hard race, a thoroughbred horse is typically getting some blood into the lungs. Okay, does that make sense to you guys? Okay, so we're under selection to have this very thin structure, but that also means there's a trade-off here. That means you have less resistance to damage. So I asked you guys, what would happen if you have particles? So when we talk about dust particles, pollution, those particles down in the range of 2.5 microns, the very smallest ones they typically measure. So a 2.5 micron particle might be something like this. Nope, that's too big, sorry. Let me try again. A 2.5 micron particle might be something like this. And one problem is they're often sharp, not always. So in your lungs, where you get these particles, you have wandering macrophages. Those macrophages will engulf particles. And normally, of course, those particles that get in are organic material. And a macrophage can often actually digest it. If it's a bacterium or something like that, or a virus, it can digest them. Often it's somewhat harmless material, but a lot of the dust we put into the air is not that mild. Some of it includes things that are actually toxic. They produce compounds that are toxic to cells. Others are simply sharp, or they can't be broken down. And so what happens is you get macrophages that have ingested these particles but can't break them down and sometimes are toxic. And they die. That process, dead macrophages release immune system messengers. We don't call them hormone, we call them cytokines. We'll talk about them next week. But we don't call them hormones because they just have local effects. But what they'll do is they will attract more macrophages. And eventually, if there's enough of them, they start stimulating fibroblasts, which are repair cells, secreting lots and lots of collagen. Okay, scar tissue. Everywhere you have a thick lump from a scar, that scar tissue, fibroblasts produce massive amounts of collagen to repair this break in your skin. And the result is you have this thick, tough area. That's good, but it's not doing what it used to. Okay? Those fibroblasts are just producing collagen. So the normal function is now gone. So good thing to start producing okay, good thing, bad thing. Okay, not a good thing. It's doing repairs, but one problem with these particles is that with enough of them, your body can't keep up. So when there are really high particle counts, hospital admissions go up, death rates go up, especially for people who are a little bit compromised. So death rates, especially for infants and for the aged, go up. They overwhelm the system. And over time, you can also have, if some of the damage is bad enough, you can have permanent damage to different parts of your lungs. Okay. So are the fibroblasts already located there? Do they move in? I don't know in the lungs specifically. In most tissues, the fibroblasts are there as a stem cell. And under the right conditions, they're triggered to replicate and start producing massive amounts of collagen. Okay, so animals can decrease path lengths, and I've just shown you how thin the capillaries can be. These are, again, about the thinnest cells in your body in order to minimize the path length. Animals can use unidirectional flow of the external medium across epithelia. So if you keep bringing in fresh oxygen and just have it go in one direction, you keep bringing in fresh oxygen, so that will increase the diffusion rates because you have a higher concentration gradient. They can use countercurrent exchange, which we'll talk about in a moment, and they can bind gases to transport molecules. Bind gases to transport molecules, hemoglobin. Okay. You hold 20 times the amount of oxygen in your blood because of hemoglobin than you would if it was just plain water. Okay, 20 times as much. You actually hold about 200 mils of oxygen per liter of your blood. And that's because 95% of it is bound to hemoglobin. Okay, so let's talk a bit more about the different systems that animals use for gas exchange. So I just talked about one, just lots of blood vessels right underneath the surface of the animal in an earthworm. Insects use a tracheal system. And the tracheal system has spiracles that open to the outside. And then inside, these tubes and then branches. It's called the tracheal system. Same root as your trachea. And it branches down to really tiny, what are called air capillaries. Or actually, in these guys, more often I'll use terminal tracheoles. It's more common in insects. So branches down to really fine structures. Here's an image from your textbook showing spiracles. Okay, showing spiracles here. So these are the openings to the outside. Often insects have around 9 of them on each side. And then opening into this system, and they're not showing all the little branches. If we looked at an electron micrograph, this is an insect muscle. So here's insect muscle, and you're looking at slices through the cells, and all the little dots aligned here together are myosins. Okay, so these are bundles of myosin, not individual molecules, but bundles of myosin. You can see how well they're arranged. There would be actin between them, which is not showing up very easily because the actin's even thinner. And here are the tracheoles. So here's one that's cut lengthwise. You can see it's a little bit ribbed. And here's one cut straight through in cross-section. Here's another one. Each of these is a cell. Notice the tracheoles are no further than about 3 or so cells from every individual cell. They're within about 50 microns, generally 20 to 50 microns from every cell. So there's oxygen near every cell in an insect. Okay. So insects don't need to send oxygen through their circulatory system because they're doing it through this tracheal system. Now that costs something. It costs resources in order to send these little passageways next to every single cell. It's probably one of the things that limits insect size. You probably don't need to worry about a wasp the size of you are flying down on you, swooping down and trying to sting you, because this, for much of its length, relies on diffusion through air, no bulk flow. So there's no bulk flow in these terminal tracheoles. There can be bulk flow in the biggest vessels. Some insects just have a rigid, the entire system is just rigid, and oxygen moves in through diffusion through air. It's a lot faster than diffusion through water, but it's still not as fast as an insect needs if it needs to be big. Really active insects actually expand and contract their bigger tracheal. So just like you expand and contract your lung, they use muscles to expand and contract. So they pull air in, fresh air in, and then they send it back out. And it goes by diffusion through these terminal branches. Insects have a lot of different varieties of doing it, and in some cases, they've lost the external tracheal. So aquatic insects, instead of having the spiracles with allowing oxygen in, they still have a hollow air-filled area, but there are branches in little gills, external gills, that pick up oxygen from the air. And then there's some really exciting ones that actually have the branches go into the anus, and they take a bubble of air into their anus. They're basically breathing through their anus, which always is a weird image for me. Applied to humans, it's particularly weird. Gives you a whole new perspective. Okay, for efficient transfer, most efficient transfer, you need countercurrent exchange. That's what fish do. So here's a trout and a gill arch. So they have 4 gill arches. Each gill arch has blood vessels going through the arch. So each gill arch is like this. So blood's going through here, and there's a gill arch, another one behind it, another one behind it, another one behind it. Each gill arch has these branches, okay, gill filaments going on each side, and the water passes across these branches. If you do a blow-up of these branches, what you see is something that looks like something flat with half pancakes sticking up from it, okay? So, like half a pancake, the blood goes in, moves through spaces or capillaries in these lamellae, each of these little half pancakes, and then heads back to the body, having picked up oxygen. Now, there's a problem with your textbook. They got the figure backwards. Okay. So the textbook says oxygen-rich blood is coming here, going across, and then somehow this is oxygen-poor blood, like the gill has given up oxygen to the water. Makes no sense. So this is oxygen-depleted blood. So here's what your book said. I put in much bigger text, oxygen-depleted blood. Should be blue. And then this is the oxygen-rich blood coming back. That's a countercurrent exchange system. The water is coming this way. Blood is going this way. Okay? So remember I talked about countercurrent exchange systems and some guidelines to make sure you understand them. I think in every year I've had a countercurrent exchange question on the final exam. So it would shock me and should shock you if you don't see one. Okay? And so you always want to have a source that is high in whatever it is that's being transferred, and a sink which is low. If it's a counter-current exchange system, check. Are they at opposite ends? Okay? If they're not at opposite ends, it's not a counter-current exchange system. So the source is coming from one side, and the sink is coming from the other side. The source is going to lose oxygen, or whatever, temperature, whatever it is. And if your source is not losing, then it's not a source. And if your sink is not gaining, it's not a source, it's not a sink. Okay? And then the last thing you have to check is oxygen always moving down a gradient, or whatever it is. Is heat always moving from an area of higher heat to lower heat? Is oxygen or whatever else you're transferring always moving? Okay? So here's your source, water coming in at, say, 160 millimeters of mercury, partial pressure, and it encounters blood that's a little lower. Not that much lower, because the sink has already picked up a lot of oxygen. Notice it's picked up oxygen all the way along here. And so it might be 140. Here, the source, the water, has lost some of its oxygen, so maybe it's at 120, but the sink is still lower. It's coming from this area that has even less, so it's at 110. And this whole length, you're going to have oxygen transferred from the area of higher amounts to the area of lower amounts. Okay? So, source and sink at opposite ends, okay? And the source needs to lose, the sink needs to gain, and whatever it is always has to go from an area of higher to lower amounts, right? I'm making a big deal about this because in past years, roughly 35% of the class gets part of this messed up. I think the reason is it seems very straightforward when you look at it here. And then when you have to remember it and go through it on the exam, if you haven't practiced it, it doesn't stick. So I'm hoping for 100% this year. Source, sink. I got a few light, faint chuckles. This is remarkable. Okay, so that's gills. We talked a bit about mammalian lungs, so I wanted to familiarize you with a bit of this. Most of you, in fact, you all should have seen this in a high school class. But you've got an area where air is coming in, and we have what is called a tidal lung. So air comes in and out like the tides. So air comes in. It comes in because you contract your diaphragm. Here's this muscle, very thin muscle here, and when it contracts, it flattens out. So contracts, it flattens out. That creates a little bit lower pressure in your lung than in the air outside. So air moves in, fills it up. Moves down your trachea and bronchi and bronchioles, and then finally down into these terminal regions, the alveoli. And the alveoli, I already showed you a picture. Here's this opening into an alveolus. Here's all these blood vessels, this network of blood vessels, and it's picking up oxygen and getting rid of carbon dioxide. It's an inefficient system. The problem is, first of all, we have some dead space. So we have some area here where you can't do any extraction. As you deplete, you bring in air and you mix it with the air that's already partly depleted. The best you get inside here, at best, you get around 120 millimeters of mercury. Okay, somewhere in there, maybe a bit higher than that. If you're breathing fast, it's higher than that. And when you're not working hard, right now, the air you're breathing out is actually not far off 120. That's why mouth-to-mouth resuscitation works. Okay, my videos, I made it a little more extreme. And so the air that you're trying to pick up, first of all, it's already lower than what was in the air, and then you just get passive exchange back and forth. So in the blood, the best you get, I think it's in a lower diagram down here. So for oxygen, The best you get in your systemic capillaries in this diagram is around 100 millimeters mercury of blood. Okay. That's sort of averaging between the 120, 130 you can get when you just inhaled, but then lower amounts when you've exhaled a bit, and of course you don't get perfect transfer. You never get all the oxygen that's there. So somewhere around 100. Elsewhere I've read 105, maybe even if you're breathing fast, 110, 120. But somewhere around here is the best. 100's a reasonable one to remember. So you have only a third the oxygen that was in the air. A fish can get almost all the oxygen that's in the water going through the gills because of that countercurrent exchange system. So they can get to 150 millimeters of mercury partial pressure of oxygen, okay, because of countercurrent exchange. Because there's so less oxygen in water, they practically have to do it. If you're going to be an active animal, you have to do it. And you might wonder, why is it that marine mammals have not evolved gills so that they don't have to breathe, come to the surface and breathe all the time? Well, the problem with gills is there's so much less oxygen in water. Marine mammals have such a high metabolic rate that they couldn't do it. They would need a huge gill. Gill would have to be roughly 10, 20 times the size of a fish gill to get enough oxygen. So imagine a whale that was, I don't know, 40% gills. Wouldn't work. So they rely on air, which has much more oxygen. Okay, so you breathe in, you breathe out. Remember all those dust particles I talked about? Well, you have systems to get rid of them. Now, you can't put cilia with lots of mucus in your alveoli because that would increase the path length. It defeats the purpose. But all the air getting down to your alveoli has to pass in all the bronchi, so the bronchus and the bronchioles, all those branches going down, those cells are lined with cilia and they're secreting mucus. Okay, for Friday, you're going to be watching a video on the manufacture of mucus, which is a highly underappreciated topic. Especially in this season of colds, I want you to appreciate mucus more. Anyway, mucus with cilia, and these cilia are constantly beating, moving the mucus up toward the back of your throat and out into your throat to get rid of it. So the mucus is trapping a lot of dust particles, a lot of bacteria. Most of the dust, most of the bacteria, most of the viruses you breathe in get trapped and get moved back out. The ones that make it all the way to the alveoli, those are the problem. And there's a scale bar here, so that's 2 microns. So these cilia are maybe 6 microns. Okay, remember a typical cell is about 10 to 20 microns. Okay. Okay, I think let's have you do a countercurrent exchange system. So sketch for yourself or explain to your neighbor a countercurrent exchange system in a gill. Okay, countercurrent exchange in a gill. Okay, see if you can close your eyes and picture all the steps. Picture the amounts, picture the movements. So I gave you a homework problem in which somebody diabolical had changed the direction of blood flow through a gill so that instead of going in this direction, the blood is going in this direction. And I wanted you to think about what's going to be different. So you can put the same numbers here if you want, but what I really want you to do, I'll tell you, if you put the same numbers there, it's wrong. So what would you actually expect at this far end if we did this? So this is now 20, right? The 20 that was coming in at the other end is now coming in here. So are we going to get gas exchange? Okay, is it going to be faster or slower than the gas exchange before? Faster, bigger concentration gradient. Okay, so maybe we're going to have, instead of only a tenfold difference, maybe we get to 40 here. And so further along, where it was 120, is it still going to be 120? It's going to be lower or higher? So maybe it's 100. And so maybe this is up to, let's call it 80. Okay? And then a little further, is it still as fast, gas exchange? Not especially fast. Maybe by here we get to 90 and 90, and now what happens? It just stops. Okay, does that make sense? Okay. That would be a co-current. Okay, that would be a co-current exchange. And less efficient. Just by having opposite directions of flow, you wind up with much more efficient transfer. Okay, any questions before we talk about bird lungs, which are a little more complicated yet? Okay, bird lungs. Bird lungs are complicated structures. These are already simplified. But we simplify them in order to make sense of how they work. It's taken a long time and we still don't fully understand them. But I'll give you a sense of what we think we know now. So this was a video you guys watched. I hope you have practiced drawing bird lungs, the Mickey Mouse ears. And parabronchi going through here, and the parabronchi have their own little air capillaries going off. Okay, parabronchi, you have bronchi. These are sort of like bronchi, parabronchi. Okay, inhale. I'm going to go through this quickly because it's in the video. Inhale, that's closed. And all of these sacs are expanding. That means air is being pulled out of the parabronchi. And so we have one-way flow of air through the parabronchi. Air is also being pulled in here, which means air is going into the bird mouth. So when the bird is inhaling, air is moving in one direction to the parabronchi. Do it again for the exhale. Mickey Mouse ears. I can't tell you how many more people get this diagram correct when I just use the phrase Mickey Mouse ears. I don't know what this thing is in the middle. A hat? Okay. Again, the parabronchi. Now, on the exhale, this is closed. Okay. And all the air sacs, there's actually more than two, contract. Well, if these back air sacs are contracting, the only way the air can go is through the parabronchi. And so it has to be coming out this end, and all this depleted air is going out. So on the exhale, air is moving in the same direction through the parabronchi. Doesn't matter whether the bird is inhaling or exhaling. Air is moving in one direction through the parabronchi. So they get single uniform direction of flow. Which way would you predict the blood is going? On here, left to right or right to left? Okay, you would predict countercurrent. Now, it turns out the distances are too long, the anatomical structure is a little more complex. They can't manage a true countercurrent. It's often called cross current. I tend to call it near countercurrent. And so a bird can get partial pressures of oxygen in its lungs that are 30, 40, 50 millimeters of mercury, in its blood, higher than you can. That means they can get more oxygen out of the air. And so you can have that great picture. Here's Mount Everest. Here's our poor, exhausted person. And here's a bird. Flying overhead. High energy Himalayan goose flying overhead of this poor climber on Mount Everest. And if they're not acclimated, our climber on Mount Everest is actually dead. Okay, so more efficient. So, again, we are a jury-rigged system. Our system is good enough, but a one-way flow of air through the lungs would be better. If you are an athlete and you have ever run out of breath, run low of breath, running down a field or swimming or whatever, you want the respiratory system of a chicken. You would be able to do 40% more, maybe 50% more, just by having the respiratory system of a chicken. Yeah, Sarah. So I've never found out what they are. I presume they're just sphincters. They're just a ring of circular muscle, because these are not really high pressures, so just a ring of circular muscle would close it down. But I don't know. I've never been able to track it down. So birds achieve arterial partial pressures of oxygen that approach 160. Mammals, the best we do is around 100, maybe a bit more than that. Simeon? Yeah, so do we know what causes those closures? That's what, that was Sarah's question. It's probably just a ring of circular muscle, but I've never been able to track it down. So they're low pressures. So my guess would be so low pressures that are just squeezing that with circular muscle would do it. Okay. But like I said, I've never been able to find it, so I don't know for sure that it works this way. Okay, the last thing I want to do today is talk a little bit about the use of respiratory pigments. So respiratory pigments, they're called pigments because they actually change color when you add or remove oxygen. Your blood really does turn blue when it doesn't have oxygen attached. And the oxygen comes in, binds to an iron held in a heme group. And in you, the hemoglobin is a tetramer. It's 4 proteins, each with its own heme group. So each hemoglobin will hold 4 oxygens. In other animals, it can be multimers. They can actually be hundreds of them stuck together. That tends to reduce the solute concentration because the inner ones are not in contact with water, so there's some reasons to do it differently. In you, they are packaged in red blood cells. Okay, I can tell everybody is rustling, so it must be time for me to quit. So next time we'll come back with myoglobin, oxygen binding curves, and then we'll get into digestion and mucus.